Chapter 6 Tempest in a Cereal Box. Genetically Modified Agriculture. In June 1962, Rachel Carson's book Silent Spring created a sensation when it was serialized in The New Yorker. Her terrifying claim was that pesticides were poisoning the environment, contaminating even our food. At that time, I was a consultant to John Kennedy's President's Scientific Advisory Committee, PSAC. My main brief was to look over the military's biological warfare program. So I was only too glad to be diverted by an invitation to serve on a subcommittee that would formulate the administration's response to Carson's concerns. Carson herself gave evidence, and I was impressed by her careful exposition and circumspect approach to the issues. In person, too, she was nothing like the hysterical eco-freak she was portrayed as by the pesticide industry's vested interests. An executive of the American Cyanamide Company, for instance, insisted that if man were to faithfully follow the teachings of Miss Carson, we would return to the Dark Ages, and the insects and diseases and vermin would once again inherit the earth. Monsanto, another giant pesticide producer, published a rebuttal of Silent Spring called The Desolate Year and distributed 5,000 copies free to the media. My most direct experience of the world Carson described, however, came a year later when I headed a PSAC panel looking into the threat posed to the nation's cotton crop by herbivorous insects, especially the boll weevil. Touring the cotton fields of the Mississippi Delta, West Texas, and the Central Valley of California, one could hardly fail to notice the utter dependence of cotton growers on chemical pesticides. En route to an insect research laboratory near Brownsville, Texas, our car was inadvertently doused from above by a crop duster. Here, billboards featured not the familiar Burma Shave ads, but pitches for the latest and greatest insect-killing compounds. Poisonous chemicals seemed to be a major part of life in cotton country. Whether Carson had gauged the threat accurately or not, there had to be a better way to deal with the cotton crop's six-legged enemies, then drenching huge tracts of country with chemicals. One possibility promoted by the U.S. Department of Agricultural Scientists in Brownsville was to mobilize the insect's own enemies, the polyhedral virus, for instance, which attacks the bollworm, soon to become a greater threat to cotton than the boll weevil, but such strategies proved impracticable. Back then, I could not have conceived of a solution that would involve creating plants with built-in resistance to pest insects. Such an idea would simply have seemed too good to be true. But these days, that is exactly how farmers are beating the pests, while at the same time reducing dependence on noxious chemicals. Genetic engineering has produced crop plants with onboard pest resistance. The environment is the big winner, because pesticide use is decreased, and yet, paradoxically, organizations dedicated to protecting the environment have been the most vociferous in opposing the introduction of these so-called genetically modified GM plants. As with genetic engineering in animals, the tricky first step in plant biotechnology is to get your desired piece of DNA, the helpful gene, into the plant cell, and afterwards into the plant's genome. As molecular biologists frequently discover, nature had devised a mechanism for doing this eons before biologists even thought about it. Crown gall disease results in the formation of an unattractive lumpy tumor known as a gall on the plant stem. It is caused by a common soil bacterium called Agrobacterium tumefaciens, which opportunistically infects plants where they are damaged by, say, the nibbling of a herbivorous insect. How the bacterial parasite carries out the attack is remarkable. It constructs a tunnel through which it delivers a parcel of its own genetic material into the plant cell. The parcel consists of a stretch of DNA that is carefully excised from a special plasmid and then wrapped in a protective protein coat before being shipped off through the tunnel. Once the DNA parcel is delivered, it becomes integrated as a virus's DNA would be into the host cell's DNA. Unlike a virus, however, this stretch of DNA, once lodged, does not crank out more copies of itself. Instead, it produces both plant growth hormones and specialized proteins, which serve as nutrients for the bacterium. These promote simultaneous plant cell division and bacterial growth by creating a positive feedback loop. The growth hormones cause the plant cells to multiply more rapidly, with the invasive bacterial DNA being copied at each cell division along with the host cells, so that more and more bacterial nutrients and plant growth hormones are produced. For the plant, the result of this frenzy of uncontrolled growth is a lumpy cell mass, the gall, which for the bacterium serves as a kind of factory in which the plant is coerced into producing precisely what the bacterium needs, and in ever greater quantities. As parasitic strategies go, Agrobacteriums is brilliant. It has raised the exploitation of plants to an art form. The details of Agrobacterium's parasitism were worked out during the 1970s by Mary Dell Chilton at the University of Washington in Seattle, and by Mark Van Montague and Jeff Schell at the Free University of Ghent, Belgium. At the time the recombinant DNA debate was raging at Asilomar and elsewhere, Chilton and her Seattle colleagues later noted ironically that in transferring DNA from one species to another, without the protection of a P4 containment facility, Agrobacterium was operating outside the National Institutes of Health guidelines. Chilton, Van Montague, and Schell soon were not alone in their fascination with Agrobacterium. In the early 80s, Monsanto, the same company that had condemned Rachel Carson's attack on pesticides, realized that Agrobacterium was more than just a biological oddity. Its bizarre parasitic lifestyle might hold the key to getting genes into plants. When Chilton moved from Seattle to Washington University, St. Louis, Monsanto's hometown, she found that her new neighbors took a more than passing interest in her work. Monsanto may have made its entry late in the Agrobacterium stakes, but it had the money and other resources to catch up fast. Before long, the Chilton and the Van Montague Schell laboratories were being funded by the chemical giant in return for a promise to share their